Hello everyone. So today I wanted to go over a little bit of how I handle sampling fungi for DNA sequencing. So I have my PCR plate with some tube strips in it, and I got some more tube strips back here. I have my mushrooms, I have some alcohol wipes, and I have very sharp tweezers. These are really sharp and smooth. I like to use ones like this. I find that if they have any serrations on the tips here, that materials can get caught in them, and that can lead to cross-contamination, and you can deal with it in the validation process, but it's a pain and you don't want to have to deal with it. And then I also have a Sharpie for marking up my tubes when I'm done. So basically, oh, and a spreadsheet to log my stuff. So basically what you do is you will put whatever your unique identifier is into the spreadsheet. And I like to check these and just make sure, because I use iNaturalist numbers and I want to make sure it's connected to the correct iNat number. So I usually will pop it on the observation just to make sure I'm looking at the right one. But you can also use Mushroom Observer, you can use microportal numbers, you can have your own lab codes if you want, as long as it's a unique identifier that you can use to track the specimen. And now what I want to do is I want to go in on the hymenium of this mushroom. So the hymenium being the fertile surface, right? The pores, the gills, the tubes, you know, what have you. Ascomycetes can be a little weirder to figure out where it is, but you'll figure it out. So I just want to go in the hymenium and grab a tiny piece, right? So this piece would be way too big. This is way too much material. If you put too much material in these tubes when you're sampling, the PCR can fail. It can overwhelm the PCR process. And that's not good. So realistically, we want about material like that. You probably can't even see it. It's a teeny little fragment. If you think it's too small, it's probably the right size. Like if you sit there and go, I think this piece should be bigger, it shouldn't. Make it as small as you can. So basically knock that in your tube, make sure he's in there, and close the tube strip. Wipe that off. And then I will stick this over here. And then you just go to the next one. And so normally when I am doing this, I also like to kind of catalog things in my fungarium with a unique code. That way, if I ever have to send a split off somewhere, or I have a code for it, that's a lab code, or I can find it easily in boxes. Because basically, I put it in my fungarium sheet, I put it in bags, I put them in boxes. That way they're organized, so if I do have to go back and look at something later, I can find where it is exactly. It does slow the process down slightly, but personally, I would rather just organize it while I go, so I don't forget to do it later and regret not doing it. Just like to knock everything out at once. So yeah, just grab a tiny little piece of fungi here, drop it in there. For resupinate crusts like this, any bit of the fungus is really gonna do the trick. Just a little bit. Make sure he's in there. I'm probably not going to do the whole row here, but I'll just do a couple more to kind of show you where I'm going with this. You know, and you just repeat ad nauseum. It is a bit tedious work, but I enjoy this sort of work. Not everyone does. And that's okay. But if you do want to sequence things, learning how to sample them is realistically the best, because even if you don't have all the equipment to do the PCR process yourself, at least doing this saves a ton of labor on the part of the sequencer, and it's really the biggest bottleneck. So learning how to do this properly is pretty important. It's going to save you money for sequencing. Yeah, you just kind of go down the line here and do your mushrooms. Trying to pick out interesting ones. And then for xylaria, just try to get the hymenium like this outside surface here. Just try to get a piece where you can. These little hard ones can be a bit tricky to get a small piece out of, though I don't find like the xylaria and the polypores quite as bad as I do like ascos. Ascos tend to dry very hard, and yeah, it's hard to get a sample off of them. I've bent tweezers trying to get a sample off of a tiny asco before. Yeah, I mean, you just kind of keep repeating this until you're done. So I'm not going to do the whole row here, but I will show you kind of how I mark these up. So how you mark your tubes is going to depend on who is doing your sequencing, right? You want to figure out how they want everything labeled and how they want their spreadsheet set up. In my particular case, I'm putting the row number on the left side right here, and then I'm also marking these like in a pattern for redundancy, so it's really easy to tell at a glance, you know, what row goes with where. Like this is useful, like so if someone drops a whole tube strip and these all scatter, you can easily put everything back in place where it was supposed to be, not have to worry too much about it. And then I've also written the plate number at the end here on this last tube. So this is plate 5. But yeah, that's about it. I hope that helps. Remember, if you're doing this, teeny tiny little samples. Teeny. Make them as teeny as possible. But thanks. Have a great day. Hope that was educational. Bye-bye.